the genetic polymorphism of bees, which are widespread in Ukraine today. Ukraine is the largest producer of honey in Europe and one of the largest producers of honey in the world. Ukraine has a variety of natural physiographic zones with favorable climatic conditions for producing high-quality honey. Small private apiaries in rural traditional cultural landscape settings are the base of honey production in Ukraine. Currently, the number of beekeepers in Ukraine is about 400,000, with a total of 3 million bee colonies. It is believed that 3 subspecies of honey bees are widespread in Ukraine. In Europe, there are several subspecies of Apis mellifera, which arose as a result of adaptation to local environmental conditions. Our university is located practically on the border between these 3 subspecies, Apis mellifera macedonica, Apis mellifera carnica, and Apis mellifera mellifera. And such location is very useful for our study. In the second half of the 20th century, Ukrainian steppe breed, the Carpathian breed, and dark European breed were widespread in Ukraine. Taking into account their morphological features, it is believed that these breeds probably represent 3 subspecies, Macedonica, Carnica, and Mellifera. The traditional approach to determining the subspecies and breeds of bees is based on the comparison of external features and morphometry. To describe the current distribution of bees in Ukraine, at the initial stage of our study, we also tried to use morphometric methods to determine the breeds of bees. In particular, the cubital index was calculated. For bees from breeding apiaries, as shown here, a distribution curves with one model class were routinely obtained. However, for bees from ordinary apiaries, curves with 2 model classes were usually observed. Similar results were obtained for other indices. Hence, a significant number of bees in Ukraine do not meet the standards and probably are hybrids. To clarify their origin, it is necessary to use molecular markers. Accordingly, we applied molecular methods to determine current geographic distribution of bees in Ukraine. To reach this goal, we decided to analyze the available information and identify genomic regions suitable for discrimination of local breeds. When we have sequenced and compared selected genomic regions in reference samples of bees of known origin and bees from Ukrainian local populations. In our study, we choose mitochondrial molecular markers. Mitochondrial DNA contains only 37 genes. It is maternally inherited and is identical in all bees in the colony. Mitochondrial DNA is widely used in molecular taxonomy. Especially, cytochrome oxidase genes CO1 and CO2 are widely used in molecular taxonomy of insects. Now, mitochondrial DNA is sequenced for many accessions of Apis mellifera, for several subspecies like Carnica, Caucasica, mellifera, etc., etc., but unfortunately not for Macedonica, which is present in Ukraine. The CO1 and CO2 genes are very similar in the Apis mellifera subspecies. The level of sequence similarity between closely related subspecies amount to 98, even 99%. However, the genes contain specific base substitutions, so-called SNPs, that can be used to distinguish subspecies. At the beginning of our experiments, there was practically no information about the mitochondrial DNA in Ukrainian bees. To clarify taxonomic position of Ukrainian bees, samples from different regions of Ukraine were compared with reference samples and sequences from GenBank. As reference samples, we have used several lines of Carnica from Austria, a line of Macedonica from Greece, and 4 lines of Carpathian bees from Ukraine, from the western parts of Ukraine. We used standard experimental procedure, DNA extraction, PCR amplification and sequencing of PCR products, identification of specific SNPs, and genotyping of bees from different regions. 3 pairs of primers, they are used to amplify different parts of CO1 and CO2 genes. And here are our results. You see the sequences of reference samples, Carnica lines from Austria, Ukrainian Carpathian bees, Macedonica line from Greece, and also samples from different regions of Ukraine. As you see, application of the first pair of primers allows distinguishing of Carpatica plus Cardica and Macedonica. However, we were unable to discriminate Carpatica and Cardica bees. Application of the second pair of primers did not lead to discrimination of Carpatica, Carnica, and Macedonica. And finally, the use of the third pair of primers allows to distinguish Macedonica. Specific SNPs are shown here. And 2 groups of Cardica. Carnica 1, which includes several lines from Austria. Always lines contain 4C here for cytosine residues. And Cardica 2 plus Carpatica bees. They contain here only 3 residues of cytosine. So, using the obtained sequences, we built a map reflecting current distribution of subspecies in Ukraine. According to traditional views, Carpatica should be present only here, in western region. There's Macedonica should be present in central and eastern regions, and mellifera should be present here in northern part. But mellifera was not found at all. Thus, the current distribution of breeds and subspecies does not correspond to the traditional zoning. And now conclusions. Traditional morphometric analysis is insufficient to determine the subspecies and breeds of Apis mellifera. Comparison of sequences of mitochondrial genes CO1 and CO2 and the spatial region between them shows that mutations present in these regions allow discrimination of the subspecies Carnica, Macedonica, and mellifera. Molecular genotyping revealed that at least 2 subspecies of honeybees occur in Ukraine, Carnica and Macedonica. These subspecies are represented by the Carpathian and Ukrainian stem breeds. Apis mellifera was not found in the sampled populations. Widespread practice of importing genetic material of Apis mellifera from different regions of Ukraine leads to uncontrolled hybridization between them and represents a threat to the conservation of aboriginal breeds of honey bees.